Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast. Ozcast. Simply head to ozcastnetwork.com for details.
Life is uncertain. It's okay to feel stressed, anxious, worried, or frustrated. CalHope can help. Access CalHope's free and secure mental health resources. Call 833-317-4673 or live chat at calhope.org.
Dr. Travis Brown, what is This Medical Life all about?
This is the pursuit of knowledge as we learn about diseases from the ancient times to the present day. These are the stories of medicine.
Dr. Travis Brown, as I saw this show coming up in the show notes about Q fever, I can't help but get Peggy Lee's song Fever out of my head. Do you have that in your head now?
No, it's a little bit older for me than that one.
And we're off and racing. All right. So
yes.
Tell us about Q fever.
Well, look, Q fever is interesting. It's one of those ones. It's a disease that I've always wondered how it was named, and we're actually going to get a little bit into that today as to where the name came from, Q fever. And the story actually starts in 1934, and this happened oddly enough in Brisbane, Queensland, Australia, where we have three physicians, and they're having a discussion. Now, each of them separately has encountered a puzzling illness, and that workers were all from an abattoir, and they presented with just feeling unwell and this fever. Now, each of them had done some investigations that they would typically say would be the standard of the time, but none of them were able to find a cause. So they made an agreement. They said, we're going to admit the next patient that we come across who has this, mystery fever, and we will investigate them, do a full workup, and actually find the cause. The problem is they did this, but they were still unable to find any cause for this illness, for this, the person was an abattoir worker as well. And so that's what they called this mysterious illness. They said abattoir fever. Now, in 1935, the Queensland meat industry decided that they needed to find out a little bit more about this, because you just can't have workers getting unwell. No one knows what it is. Is this, is it real? What is it? Is it, serious? Do they have to manage it in a different way?
And I suppose the other thing is, could that lead to contamination in the food chain as well?
Yeah, well, your customers, is it a problem for customers? But is it your whole workforce? Is this just going to, take everyone out? Mm-hmm. And so they actually asked the health department to investigate. And they got a pathologist who was there. His name was Dr. Edward H. Derrick. And he was tasked with finding out what is the cause for this illness that their workers are getting. Now, for the background for this, the meat industry in 1931 had centralised their slaughterhouses, and they had closed some peripheral ones, and they were putting it into a centralised slaughterhouse, and they were then finding that these illnesses were just cropping up. And since 1933, the plant had 800 workers. There'd been over 20 cases of this mystery illness over the course of about 30 months. And so people coming away, getting unwell, it caused a bit of consternation for them. And so Edward Derrick started to investigate. And when people got sick, he started to do the tests that he could do. Now, he did blood cultures, they were negative. He did serology, that was negative. He even went to the point where he investigated animals. And, he got out from my reading, he got out the veterinary textbook and went through it all. And then tried to find, is there any signs coming from the animals that they're sick and this is somehow passing it on to the workers, but he couldn't find any illness in the animals.
Just a thought, at the moment there are some GPs listening say, oh, for those days when we didn't have the workload and we could have gone off and done a deep dive.
Well, he was employed by the health department and they had tasked him with all of this. So this was like a secondment for him. Yep. So now they went, and then, so the next area is he couldn't find anything with the patients. He couldn't see anything with the animals. So he went to the abattoir and he investigated the abattoir. Is there a contaminating source there? And he looked and he saw, a few things he noted. Well, the floor was constantly wet. Either blood and guts, unfortunately, or had to be washed down with water. So it was always wet, that environment. The dust was, contaminated, the water supply. And in the area, it's unfortunate, but he noticed that cockroaches flourished throughout the abattoir. But he didn't find any significant findings. Like, they were noted, but it didn't seem to be the cause of this illness. So it caused a bit of confusion as well with this investigation, because there was no consistency. The people who were getting sick, there was no specific animal that they were all related to. The people who became ill didn't have a specific job. They could be anything from a yardsman to a person responsible for killing the animals to someone who was a cleaner. And so there was no consistency. Some of the patients had contact with cattle, some with sheep, some with pigs. And so nothing seemed to link all of these people who were getting sick. He noticed the people who actually got the largest incidence of the disease was from the tank house. And the tank house is where all the offal goes, and they boiled it down to make fertilizer. And so I can imagine what that's like.
Oh, gee.
Yeah, but that was where he sort of said, yeah, that's the most of the fertilizer that's going to, is that something to do with that? But it didn't seem to link. And so he recognized that this disease was actually similar to another disease. It was called murine typhus. Now, this is a disease that is associated with rickettsia. Now, rickettsia is an organism that what we call is an obligate intracellular organism. It means that they're organisms that have to live inside another cell. And so these are actually really hard organisms to grow, because you need a cell for the environment to be right for it to thrive. And so we don't actually grow cells like that in a routine. We actually put it on media. And so to do this, you would actually have to have like what they were doing with viruses and put it into a hen's egg, a fertilized hen's egg, so it could grow within a cell. But he was looking at this and saying, look, this may be with rickettsia, which spreads by ticks and lice and mites, maybe this is the life cycle that it has. Now, the one for this rickettsia disease, the murine typhus, it has a life cycle where an infected animal can be like a rat or a cat or an opossum, gets bitten by a flea, the flea catches the rickettsia, goes and bites another animal, and then that's the life cycle that it goes through.
The circle of life.
That's right. Well, and when that flea who's infected with the rickettsia bites a human, we're actually not a typical host for it. And so what happens is the flea bites our skin, it breaks the skin, causes a bit of irritation. The flea then, no other way to say this, the flea poos, it's called flea dirt, and we scratch and we rub that into the wound, and that's how we get infected with that rickettsia.
It's a beautiful thing.
And so you have this person scratching, rubbing that in, we get the rickettsial infection. And this is where that cycle. So he's going, well, maybe this illness, this fever is very similar to this. And so he started looking, is there another vector? Is this a tick vector? Is there something that's coming through? Is there fleas about? It was interesting because this disease is similar in that it doesn't go from person to person. So when someone had a fever, another person wouldn't catch it from them. And so that was really interesting. So to test this theory, Dr. Derrick used guinea pigs, and he was worried that what would happen was maybe the pathogen's really fragile. Maybe, because we can't isolate it, we can't find it. It's really fragile. It's really difficult to either culture or survive. So I need to be careful with it. So at the bedside of, a young stockman who came in with his parents who had this, abattoir fever. And so Dr. Derrick thought, okay, well, what I'll do is I will inject the blood from the young stockman into the guinea pig, and then I'll be able to culture it. The problem is he injected the blood from the young stockman into the guinea pig, and the guinea pig suddenly died.
Oh, whoa.
And the quote I like, he said, is, "To the consternation of the patient and his parents." And so...
And the guinea pig, one would think, too.
Well, and so everyone started getting worried. But despite this, it would just seem to be an unfortunate event. So he continued this experiment with guinea pigs. And it turns out the organism's actually really hardy. It's really resistant. Now, he learned that the incubation time was about 1 to 2 weeks. And he was able to pass this infectious agent. So it had to be infectious. He could pass it through blood from an infected person or the urine of an infected person. And if he got a guinea pig infected, he was able to ground up the spleen or hepatic tissue and then pass it on to another guinea pig. So it was quite widespread. The other thing that he noticed was that if a guinea pig had the illness before, they wouldn't get it again. So there was immunity with it. And so he was able to use this information to then test the patient. He was actually able to test for abattoir fever, and he would get their blood and inject it into 2 guinea pigs. And one was naive to the illness, hadn't had it before, and the other had had it before. And if the guinea pig who hadn't had it before got a fever and the other one didn't, then that was positive for this, quote, abattoir fever, and they had the illness and the infection. But the problem with this was it took about 2 to 4 weeks to complete. And so he still didn't know what the infectious agent was. You could pass it on, but he didn't know what it was. And even to the point of he knew that it could even survive in liver tissue that had been refrigerated for months. So this was quite a, hardy organism. He tested pigs, sheep, bulls, wild rats, lice. He couldn't find anything. So he thought, this must be a virus. And so what he ended up doing was sending a liver sample to who turned out to be quite a famous virologist, even ended up winning Nobel Prize, decades later for immunological studies, so unrelated. But his name was F. McFarlane Burnetti. And he got this sample and he put it into chick embryos, but he couldn't find any virus. He was able to confirm that you could infect mice. And from the spleen of the mice, he didn't find a virus, but he found large cells with minute intracellular coccobacilli. So he was actually able to find organisms that had been passed through. So the interesting thing was around the same time in the United States, in 1935, the US Public Health Service was investigating Rocky Mountain spotted fever, which is a rickettsial disease caused by Rickettsia rickettsii, which again is a tick-borne disease. And 2 microbiologists, one named Gordon Davis and later Harold Cox, were hoping to develop a vaccine for this disease. And so what they did is they went out and collected, a group of them went and collected a whole bunch of ticks out of a place near Missoula, and they called the 9 Mile Creek. And using guinea pigs, they were going to get these ticks, infect the guinea pigs, and then hope to create a vaccine. What they found was the guinea pigs were getting fevers and then were able to infect other guinea pigs with it. But it wasn't what they were expecting. They were expecting it to be the rickettsial disease. And it turned out that this was a mysterious fever. He ended up saying, oh, this must be, he called it 9 Mile Creek fever because they didn't know what it was. And when they looked down the microscope, they saw these intracellular coccobacilli.
Aha.
And they were able to link it. And they said, oh, they were even able to grow it in fertilized hens' eggs, which produce huge numbers of these organisms. And so it's so interesting how it happens around the same time that they come across similar disease in two different countries. And, he called it 9 Mile Creek fever, whereas Derek was actually having his naming issues of his own. The interesting thing was, people kept on calling it abattoir fever. Well, the meat industry didn't like that because, it was associated with their product. And also, not only, it wasn't just abattoir workers who got it. So he thought, why don't we call it Queensland rickettsial disease? But then the tourism people piped up and said, well, we don't want it to, stop people coming here. He then thought, well, why don't I call it X fever? But that was already being used by a different disease that would later be called Murray Valley fever. So he called it Q fever out of query fever because they didn't know what it was at the time. And the name stuck. He published an article in 1937 with the patients who had fever and intense headache, muscle aches. And then he was able to work out, and the two groups learned of each other. Derek sent a sample across to the United States, and they confirmed that this was an identical organism. And so there were several reiterations of the name, but what ended up happening was this organism was called after Harold Cox and F. McFarland Burnett. It was given the names Coxiella Burnetti.
What an intrepid group of researchers. We could have done with them in Wuhan a few years ago, just quietly. Dr. Travis Brown, I've checked myself for ticks. I'm clear. What have we got to look at now?
So just looking at the organism itself. So what do we know about Coxiella burnetti? So this is it's a small organism. It is a gram-negative rod. So that's what we stain it with, but it actually stains better with a Gimenez stain. And look, it is an intracellular organism. So it actually grows within a cell. It grows within a small vacuole. It's called a parasitophorus, a vacuole. And this is why they're so hard to grow, because you need a really specific environment. It does excellent in that environment, but just on plates, it can actually be quite difficult. And it also is a mechanism to hide from the immune system because it's within a cell in its own little vacuole. And what do we know? Look, it's closely related to Legionella. And this is, Legionella in air conditioners and things like that it can spread. And so how do we know? Where does it come from? It actually comes from infected animals, and it goes into their urine, goes into their feces, and it goes into their milk. It also particularly goes into their birth products like the placenta and amniotic fluid. The interesting thing about it is it has what they call a small cell variant, an SCV, that acts like a spore. So a spore is sort of a non-vegetative state, meaning that it can't grow, but it can survive long times. And if it does get the right nutrients, if it does get the right environment, it can then become the bacteria and divide and grow. And they've suggested that this SCV can survive in soil and in hay and in dust. It can last for years, and they've even suggested maybe decades. And so the lab environment is actually really interesting because there's been numerous times where it's been infected the lab people who are working with it. It's really highly contagious when working with it. And so we need even a specialized center to grow it. BSL-3 is actually sort of the requirement for it. But, if we look back in 1937, even Burnette's lab, workers became ill. In Derrick's lab, workers became ill. There was even the NIH in Washington, D.C. They got 15 cases and one person died as a result. Oh. So it's actually we have to be very cautious working with it. We have to be very, very sensitive to what we put it in, work in fume hoods. It has to be sealed off. And look, the amazing thing about this is the animals that it can be involved in. It can be goats, cattle, sheep, rats. In Australia, it can be in kangaroos, wombats, bandicoots, and numerous tick species. So the amazing thing is it's endemic to every country except for New Zealand.
Oh, they've got natural ability to fight it off.
It's just interesting that New Zealand has that as well. But we do know that pasteurization kills the organism. So even though it's passed in milk, pasteurization does kill it. And the really interesting thing is the majority of the animal infections appear to be asymptomatic. So when Derrick went looking, he wouldn't have been able to find it. You wouldn't know that an animal is infected with it. And the infectious dose, they've estimated to be really low. To almost 1 to 10 viable cells can cause human illness. So that's a remarkably low amount.
Yeah, that's nothing.
Essentially, yeah. And that's why it's just incredible that it can actually take hold. And this is why it can be an issue with people who are immunosuppressed or pregnant. And looking at this from a clinical perspective, there are 3 clinical syndromes that we, want to be aware of. There's acute, there's chronic, and there's something called post-Q fever fatigue syndrome. So I'll go through one of them each at a time. And so the interesting thing is the acute is about 60% of people and patients are asymptomatic. And so even in us, it may not show as a disease. If they do get symptoms, it's pretty much flu-like illness. So, fevers, chills, fatigue, muscle aches and pains, an associated headache. And so that takes us to the chronic infection. Now, chronic infection, it begins to involve organs, particularly cardiovascular seems to be quite susceptible, and especially people who have had congenital abnormalities. So that's a significant finding if it's ever encountered. Other systems that get involved are the gastrointestinal, so we can have things like chronic granulomatous hepatitis or diarrhea. If there is something that goes really involved is the CNS, but it can also involve CNS, meaning brain and spinal cord, so they can get a meningitis and a meningoencephalitis. And then we've also got bone and tendons. And look, almost everything else is really rare, but that takes us to this post-Q fever fatigue syndrome. And about 20% of people who get Q fever have this, fatigue syndrome. And it doesn't matter whether it's treated or not treated. It's the majority of people who get this are previously healthy. They don't have any comorbidities, no psychological problems, but they can get these symptoms like nausea and headache, night sweats, myalgias and arthralgias. They can have difficulty sleeping and, reduced concentration. And the problem is with this, there's no treatment for it. There's evidence that one of the treatments for Q fever in the acute phase and be it the chronic phase is the doxycycline, so an antibiotic. But there's no evidence extended doxycycline has any beneficial effect of this post-Q fever syndrome. So that's what we know about the organism and the clinical manifestations. But I think we need to get a guest in to help us explain a little bit more about the applications of all this.
Look, let's do that. And I'll just say my envy of you and your role is diminishing with each episode due to the persistence of these organisms. I mean, we've also talked about CJD, which is persistent as well in the lab. I'm quite happy doing what I'm doing. Thank you for taking one for the team for us, Dr. Travis Brown. This Medical Life. Dr. Lynn Waring is joining us. Dr. Waring completed her FRCPA in medical microbiology at Fairfield Infectious Diseases Hospital and the Royal Children's Hospital, Melbourne, as well as an infectious diseases fellowship at Stanford University Medical School in California. Dr. Waring has a strong interest in research, has worked at the Walter and Eliza Hall Institute, the WEHI, Chiron in California, and was Director of Clinical Research and Medical Affairs for Microbiology at Roche Molecular Systems. Dr. Waring's worked at Dorevitch Pathology, Princess Margaret Hospital for Children and King Edward Memorial Hospital for Women in Perth, before returning to Melbourne early in 2010 to join Melbourne Pathology as the Director of Microbiology and Immunoserology. Dr. Waring has a strong interest in education, including training new registrars, and is active in the RCPA, and we welcome you to the podcast.
Thank you, Steve.
Lynn, before Travis takes over with all his questions, I'd just like to start. How common is Q fever?
Well, it's one of the most common zoonotic infections actually in Australia, and it's probably a lot more common than we're aware of. It's a very difficult disease to diagnose, and you have to think about it really to make that diagnosis. So we're probably under-diagnosing it. There's sort of rates in about 6 per 100,000 of population in Australia, which doesn't sound a lot, but it is actually, for an uncommon illness, it's actually pretty common. And there's outbreaks occur as well. There's been quite a few quite large outbreaks throughout the world, as well as in Australia.
Now, we've been talking about the history, and it came through the abattoirs and areas like that. Who is at risk in our society?
Right. Well, interestingly, when you look at the statistics of the cases we have, men are actually more susceptible than women, and it's probably more because of their vocation rather than their protoplasm, because it's mostly you mostly acquire this from animals. So farm animals and abattoir workers is where you catch it. It's transmitted from fluids from those animals, milks and amniotic fluids and blood and all the things that you get in abattoirs that we won't go into. But also because of the innate nature of these organisms, they're very tolerant of drying, so they can exist in the environment for a very long time. And so you can actually just inhale them from being in a rural area. Even if there aren't animals present, you can also rarely you can catch it from other people, but it's actually more transmissible than we think. So although abattoir workers are often the people we target, there are a lot of other people potentially at risk. And in fact, I understand that there's been serological studies in kangaroos, even in our native Australian animals. And they can get infected by this or carry it without being infected themselves, without being symptomatic. So, it's probably a lot more around than we're aware of, I think.
Mm-hmm. Now, we're looking at the disease itself. So there's an acute phase, a chronic phase, and, post-Q fever syndrome. But is there an easy way to tell the difference between when someone's acute and then someone's gone chronic? Is there a easy distinction there?
Clinically, it's a difficult disease to diagnose because it's very non-specific. It produces a fever and flu-like illness like lots of other things do, but it tends to persist. And if you do your diagnostic tests, you find that there's nothing else coming up, then you sort of have to think about this. It also causes a hepatitis quite commonly, which is a bit unusual in some of the other things like COVID, for instance. You can actually diagnose it by serology or by molecular methods. So, in fact, if you think of the disease clinically, then you can do diagnostic tests and diagnose it. But if you don't think of it, it's very difficult because you do blood cultures, they won't be positive, they'll be negative. So although it is actually able to be cultured, most laboratories are discouraged from doing so. You have to grow it in cell cultures, which most labs don't do anyway. It doesn't grow in routine blood culture bottles. It's an obligate intracellular organism, and it's a highly pathogenic organism. It's a laboratory biosafety hazard risk. So labs don't tend to culture it except if they're a category 4 lab generally. So from the clinical perspective, the chronic symptom, the chronic phase is actually a lot more difficult. So if you know that someone's had an acute infection, then it gives you a bit of a clue that you might have the chronic infection. And most of those people would present with basically endocarditis is the most common presentation for chronic Q fever. And really you need to do an transesophageal echocardiogram and to discover that they've got endocarditis. So it should be part of the workup of that. So if you've got endocarditis and a negative blood culture, Q fever's obviously got to be on that list of things you should be doing. And you should be doing PCRs as well as serology. So I don't know if that really answers your question.
No.
The other thing is that in children, because children, of course, can be susceptible to this, they're very often asymptomatic, even in the acute phase. But like some other things, they present quite differently. They seem to have more sort of GI type symptoms, whereas adults tend to have more pneumonias, upper respiratory tract kind of symptoms, dry cough, weight loss, hepatitis. So it's like, really quite a very different clinical picture.
So you mentioned the two tests, so one with serology and PCR. When would you do one or the other? Now, serology, I'm guessing, will be mainly for patients coming on through. When is PCR going to be the most appropriate test?
Yes, so PCR needs to be done very early. The PCR off the whole blood is positive in the very early stages of the disease. And once you start patients on treatment, it becomes negative quite quickly. So you should do it early. If you do a serology and you find it's positive, you should still do the PCR though. Serology, you tend to look for a seroconversion of the IgG reaction to the different proteins, and you're looking for a seroconversion, a 4-fold increase in the IgG level. Generally, that's between an acute sample that's taken within the first week of the illness and a convalescent sample that ideally is taken somewhere around between 3 and 6 weeks after that first sample. If you don't do that, because most often people don't think of this diagnosis and they don't have that acute sample, they still should do the PCR because there is a chance it could be positive. And if it's positive, then that clinches your diagnosis. So it is always worth doing it, but it doesn't persist. So if you've got a long historical core presentation or someone who has randomly had a serology done and it's positive, it's more likely you're looking at a chronic infection and that may have not been treated and has spontaneously resolved. And that they're very important because, in fact, there's a high mortality if those people are symptomatic. Because they often will die from endocarditis and heart failure, etc., if it's not treated. So there is a chance that the PCR can be positive at that phase as well, especially if they've got endocarditis, and certainly if surgical samples are obtained, heart valves, etc., you can do PCR off the tissues. It's very specific and will make the diagnosis. There's also immunohistochemical tests, stains you can do on the tissues. So, the three things, the serology, acute convalescent serology, PCRs, and any tissue biopsies and specific immunohistochemistry strains is really the way to diagnose it. The main diagnostic thing is to think of it, because if you don't think of it, you can't make the diagnosis at all. And that's the biggest problem. Do you want me to talk about the serology? Because it is a little bit complicated.
Absolutely, because I've found the serology interpretation quite difficult because we refer out the test and then you get a phase 1 result and a phase 2 result. And can you explain to me what the actual difference is between those two?
Right. So, and in fact, hopefully in the future, we're going to move a little bit away from that too. Because the other thing is that we're doing enzyme immunoassays that don't look at the phases. So they're just IgGs and IgMs. So you can get quite a complicated report. Often the phase proteins are done through a complement fixation test results. And the phase 1 is the virulent part of the microorganism. They're not actually, they're intracellular obligate gram-negative bacilli. They're actually bacteria, but they're slightly defective bacteria. So we sort of think of them like that. So the phase 1 is the whole protein, and that's present. You get antibodies to that much later than you do to the compartmental, the partial proteins, which is the phase 2. Now, I don't know why they will call the other way around from how they develop in humans. I do seem to remember in the midst of my foggy memory somewhere that there was some explanation for how that came about, but I do not remember why we discovered the phase 2 after, that occurs before the phase 1. But it's obviously just designed there to confuse everybody. But phase 2 is what you find first, which is the partial antigens to the partial antibodies to the partial antigen, and then subsequently later you get the antibodies to the phase 1, and right. The IgG occurs. It's mostly IgG you want to see, because IgM is a very cross-reactive. Q fever particularly is quite immunogenic. Some infections are like that, and they can cause a lot of cross-reacting antibodies that cause a lot of false positive IgM. So you will see that too. So if you get a positive IgM, be considered in your approach to that. Take it on its value, on its face value. Do another sample, because right. It may be that you're very early, or that it's a false positive. And if it is truly Q fever, the IgG will become positive. And if it's weakly positive, so something like 101 in 128, that's a titer. It's just a doubling dilution titer. But when they seroconvert, they go up to the thousands. So you go from, 128 sounds high, but it isn't really high, up to like over 1,024. So that's a clear seroconversion. That's really how you clinch a diagnosis in the acute setting. And that's to the phase 2. And then sometimes by the time you get that seroconversion, you start to see the phase 1 IgG response. So you get the shift over time. Yeah.
Okay. The complexity, often a lot of the time, Q fever may not be the first diagnosis people think about, and it comes up over time. With treatment for this, is any time, if someone's either getting over it, is treatment appropriate? Is it not? What's the best way to sort of approach treatment for Q fever?
Well, it's really very, very simple. So it can be problematic, though. The ideal treatment is with doxycycline, which has got some problems attached to it. Obviously, during pregnancy, you try to avoid using doxycycline because it stains the developing teeth and children. And it stains the bones, etc., and you can have other reactions to it. There is an alternative of cotrimoxazole. It's not thought to be as effective. But if you don't treat someone with acute Q fever and they develop chronic Q fever, it's got a high fatality rate. So you really do want to treat this. So if you think about it, you need to do the diagnostic tests. And you don't wait for the tests to treat. You start treatment if you're really you're very concerned about it. Certainly if you've got someone with a culture negative endocarditis, you don't want to wait. You want to go on and treat.
Okay. Now, there is a vaccine for Coxiella Burnetti. Who is recommended to get this?
So the vaccine is actually recommended to be given to all abattoir workers before they start working in the abattoir. And that does cause some degree of problem, because sometimes people don't do that and they start working in the abattoir and they get the vaccine, and then they do the serology and they do it all in the wrong order. And of course the serology comes up positive and you don't know. And then we have been very classic cases of people like that have actually acquired it when they first started working in the abattoir, and then they got their vaccine and it becomes very difficult. That's why the PCR is really important, because that's how you can diagnose those sort of cases.
Right. Just the last question I have, is there any tips or advice you have for GPs about Q fever?
Okay, well, I think the main advice is to think about it because it's actually not that uncommon. And we live in a dry country and we have a lot of animals that could potentially be carrying it. And people interact with those animals at a high rate. We've also got an increasingly tendency for our adults, particularly as they get older, and to become immunosuppressed. So our shift in our human condition has placed a lot more people at risk for this kind of an infection. And it is a difficult disease to make a diagnosis of, and you can't make the diagnosis if you don't test for it. But so you have to think about it to make the diagnosis. Particularly think about it if you've got someone with a fever that's persisting and an abnormal liver function tests, because they're the sort of things that are a little bit unusual, and a dry cough. So often people will think of doing mycoplasmas and those kind of tests of chlamydias and things in that kind of setting. But in fact, Q fever really should be up there too. We should be sort of thinking about that a little bit more commonly, and that you don't always have to have an animal exposure as other things. So in the right clinical setting, I think do the test, but be wary of the false positive IgMs.
On that note, Dr. Lynn Waring, thank you for taking time out of your busy day to join this Medical Live.
You're very welcome. Thank you.
This Medical Life is recorded in the Talked About Marketing Studios in Adelaide. For show notes and more information about the podcast, visit thismedicallife.com.au. You can contact the hosts via Twitter. Dr. Travis Brown is at Dr. Travis Brown. That's DR for doctor. And Steve Davis is at Steve Davis. Editing and production is by Tim Whiffen. Design is by Tom Buzenjud. This has been a PathNotes Proprietary Limited production.
Life is uncertain. It's okay to feel stressed, anxious, worried, or frustrated. CalHope can help. Access CalHope's free and secure mental health resources. Call 833-317-4673 or live chat at calhope.org.
Are you a podcaster? Maybe you've got that big idea and you're looking for a network to join. The multi-award-winning Ozcast Network can get your content to eyes and ears all over the world. Join now for the first month free, and you could be featuring this sound at the beginning of your podcast.
Ozcast.
Simply head to ozcastnetwork.com for details.